Oh my gosh. Okay. All right, so sorry about this. Ignore the objective and the essential question. I keep forgetting to change those two. I keep only changing the title, so I apologize about that. But today we are going to talk about evolution, and we'll also discuss some of the evidence that is in support of this theory of evolution. As I stated on Monday, evolution is sometimes a tricky conversation to have because some people's religious and spiritual beliefs don't allow them necessarily to agree with this theory of evolution. And that's fine. There's nothing wrong with that. But of course, this is a biology class. So we will introduce and discuss some of the evidence that does exist to support this theory of evolution. Of course, we cannot necessarily prove that this is exactly what happens or what has accounted for all of the various life forms that we see and have seen on the planet Earth over the course of thousands of years. But this is probably one of the best supported theories in science. Okay, so there's a lot of evidence across different scientific disciplines in support of this theory. Today is April 15th, 2021, and it is the 4th day of our 8th unit. I keep also forgetting to upload the videos to YouTube. So I need to go back and do that for the last few days. Spring break threw me off my game. All right. This doesn't really apply to this class. I think maybe, let me check really quickly. Only 2 people in this class have yet to take the Unit 7 exam, so most of you can ignore this. But we should all be checking Power School. We should all be looking at the progress reports that I sent out to most of you. Some of you I'm still working on. But we should all be willing to do those things to stay up to date with what we need to get turned in, especially when it comes to unit exams, because unit exams are a big deal. They are worth a lot of points. They're formal grades. And they also give you the best opportunity to better understand the material you'll need to know for the EOC. Okay, so if you are missing a test, whether it be the Unit 7 test, or even something from the first quarter. Obviously, I can't give you credit for it, but we can still do it, and maybe I can find a way to fit some credit into another assignment. So if you look at your Power School, you look at your report card or your progress reports, and you see you're missing an exam, then let's try to get that done. Okay, this was a warm-up question. This was the last warm-up question. And because we are only about 5 weeks away from the EOC exam, we need to turn our attention to reviewing heavily. We also got somewhat unfortunate news yesterday from the testing coordinator on campus who said that we might have to stop teaching on like April 15th because of the testing schedule. So if that happens, then we essentially are not going to have any dedicated time to review. So we're going to have to do it kind of as we continue to learn new material. So that's why we have to do more stuff like this. So this question was the last one in the warm-up, and it says eukaryotic autotrophs contain both chloroplasts and mitochondria within their cells. Which best describes how these two organelles are dependent on one another in these specific organisms? So test-taking strategy, and I've already written some stuff on the board from my last class, but as you all are taking tests, even including the EOC exam, you should be in the habit of taking notes, of learning as you go through the test. Many of you got personal feedback from me yesterday as it relates to your progress report, and I shared with you that your biggest area of growth is test-taking skills. Some of you are doing really well on the assignments, or at least the assignments that you turn in. But you're struggling on the exams for one reason or another. So I do want to think about some of the test-taking strategies. One of the best things that I can suggest to you all is to use the scratch paper that you're given. This is not a math class, so it's going to feel weird to like take notes as you go through the test. You're not really actually calculating anything, but you can still write things down. You can still try to define terms as you see them. And then as you go through the test, that builds up momentum, it builds up confidence, and you can use it as well as you get exposed to different questions. So I definitely encourage you to make sure you have a brain dump. You will have scratch paper and a pencil available to you for the EOC exam. You should use it. And even before you look at the answer choices, right? So here I have the question available on the screen, but I haven't shown you the answer choices. That's intentional. Sometimes it makes a lot of sense to try to predict what the answer might be before you actually look at the answer choices. Some of the answer choices are intentionally designed to confuse you. So before you even pay any attention to them, you might want to go into it having some confidence. Okay, so this actual question says, which best describes how these two organelles are dependent on one another? Obviously, you guys have already seen the answer choices if you've done the warm-up, but what do you all think? Does anybody have any thoughts about this? How are mitochondria, or that's a plural word, so how are mitochondria and chloroplasts dependent on one another? Excellent. Yeah, I couldn't have said it better myself. And I'll say it one more time just so the folks at home can hear. Sally just said that chloroplasts make glucose, and the mitochondria then take that glucose, which is basically just sugar, it's food, and they take that food and they use it to make energy. What's the process of making energy called? Does anybody remember? It sounds kind of like that. There is a cis at the end, or there's a, never mind. There is no cis, but what's it called when you're like breathing? Inhaling. Okay, that's true. I'm looking for a different word, but you're right, Paris. um, Respiration. Did you say that, Lisa? Good. Respiration. Yeah, respiration is the process of making energy. So chloroplasts make food. Mitochondria use that food for respiration to make energy. That's a great prediction about what the answer should say. And we haven't even looked at the answer choices yet. Okay, so that's what I want you guys to be in the habit of doing. Now, when we do look at the answer choices, first we want to define key terms. Eukaryotic autotrophs, we didn't really need that for the answer though. Chloroplasts, yes, we needed that. Sully was able to tell us a little bit about that. Mitochondria, we do need to know what those are. Sully told us that. And we need to understand that they are dependent on one another. Just as a reminder, what is an autotroph? And I know I kind of wrote it on the board, but what is an autotroph? What's that, Lisa? They make their own food, yeah. So autotrophs do a special process called what? Photosynthesis. They use sunlight, energy from the sun to make their own food. And then what is a eukaryotic organism? What's that? Yeah, a eukaryotic organism is more complicated. And why is it, or more complex? What makes it more complex? It has a nucleus, and as well as other organelles like chloroplasts and mitochondria. All right, so we just want to be clear on these terms. They will continue to come up. Okay, so here come the answer choices. I'm just going to show, oops. But again, Sully made an accurate prediction, and what Sully said is basically exactly what the correct answer says. The chloroplast is responsible for creating glucose through a process called photosynthesis. Then the mitochondria use that glucose, they use that cellular food to produce ATP, which is cellular energy, through a process called respiration. That is an essential dependency, a cycle. The energy then gets used again by the chloroplast to make more food and to do other cellular processes. Okay, so this is an amazing cycle that makes plants, pretty remarkable. All right, now let's take some notes. Let's get through this quickly. I'm going to play the video. The definition of evolution is something that you should write down, but it will be on the screen while the video plays.
And contributions from biology The theory of evolution doesn't tell us exactly how life began on Earth, but it helps us understand how life, once it came into existence, diversified into the many incredible forms we see now and in the fossil record. It also helps us make sense of the way in which modern creatures continue to adapt and change today. In biology, evolution can be defined as any change in the heritable traits, those are physical traits like fur color in mice, spots on the wings of butterflies, or instinctive behaviors like the way that dogs greet their friends with a sniff, within a population across generations. This definition can be a bit confusing, so let's see how it works. All healthy living things, from single-celled amoebas to flowers to dolphins, are capable of reproduction. We have children, we make copies of ourselves. We do this by duplicating our DNA and passing that DNA on to future generations. DNA is a chain-like chemical stored inside each one of your cells, which tells them how to grow and function. Your DNA contains coded information on how to build you. The information in your DNA is different than that of, say, a daffodil's DNA, which is why you look and function differently than a daffodil. The information in your DNA is slightly different than that of Elvis Presley, which is one of many reasons you don't look or act quite like he did. Single-celled amoebas and other simple creatures reproduce by copying their DNA inside their guts, moving both copies to either side of their body, splitting in two right down the middle, and then growing back to full size. If all goes well, the two new amoebas will be exact copies of each other, but in nature, things aren't always perfect. When DNA is being copied, errors can occur which modify the DNA code. This is what we call a DNA mutation. These mutations, which happen completely on accident and randomly to any part of a DNA strand, can produce variation in the body shape and function of the creature who inherits the modified DNA. In this case, our new little friend has an arm that stretches extra long. If he survives long enough to grow and reproduce, that extra stretchy arm, which is now coded for in his DNA, will be passed on to his children. Evolution. Any change in the heritable traits within a population across generations has officially occurred. Reproduction for dolphins and badgers and people is a little more complicated. We have to find ourselves a partner. When two badgers get together and, fall in love, a sperm cell from the father, which contains a copy of half of his DNA, only half, combines with the egg cell of the mother, which contains half a copy of her DNA. The result is a brand new cell with a complete set of DNA instructions, all the information needed to divide and grow up into a brand new badger. The new child matures to be similar to her parents, but also unique because she develops some traits from her mother's DNA and some from her father. Her new combination of traits can be passed on to her children, and again, evolution. Any change in the heritable traits within a population across generations has officially occurred. Besides a unique recombination of her parents' traits, she might also have developed some completely new traits of her own due to DNA mutations. Maybe extra hairy ears, for example. If she survives long enough to have kids, her DNA will combine with the DNA of her partner, and she'll pass on those extra hairy ears to at least some of her children. Again, evolution has officially occurred. So there you have it. Evolution is really pretty simple. Scientists and normal folks everywhere witness evolution happening all the time. Small changes like the ones we've seen here can add up over multiple generations to create dramatic changes. If you were to go back in time just a few thousand years, you'd find that all dogs, for example, originally evolved from an ancestral group of gray wolves. The evolution of those wolves from generation to generation was guided by humans. People were selecting wolves with the traits they liked, letting them breed, and then only keeping the puppies with the most desirable traits. As time went on, different breeders preferred dogs with different features, some selected for large size, some for small size, some for brains, others for brawn. Today, wolves have branched out into hundreds of breeds, very few of which look and behave much like their ancestors. A massive amount of observable evidence from many different fields such as genetics, chemistry, paleontology, and mathematics overwhelmingly suggests that just like all dogs share a common ancestor, all living things, me, you, pufferfish, banana trees, if you go back far enough, also share a common ancestor. We are literally related. We don't know what the first life form was or exactly how it came to be, but the simple process of reproduction with variation over billions of years looks to be responsible for all the diversity of life we see today. Now, you might be saying, wait a minute, hold on here. Isn't evolution random? To do something functional like turn a wolf into an adorable mini poodle, random evolution had to be carefully guided by an intelligent dog breeder. Researchers claim that all mammals evolved from an ancient shrew-like creature. But the difference between a shrew and an elephant is far greater than that of a wolf and a poodle. Who guided that process? Who was the breeder? In the mid-1800s, two men, Charles Darwin and Alfred Russel Wallace, independently discovered that a breeder is not necessarily needed. There is another force capable of guiding random evolution to produce order and complex function. They called it natural selection, which happens to be the entire topic of our next video. But before you move on, let's recap what we've learned so far.
So here's what I don't like about that video. It focuses too heavily on eukaryotic organisms. Prokaryotic organisms can undergo evolution too, and that's important to know. And I also don't like the fact that they make it sound like evolution happens between one generation. Like changes get passed from parents to children, and that's evolution. That's not the case. Evolution only occurs over the course of many generations. Because, of course, your parents pass on unique traits to you, your parents pass on mutations to you, but that does not constitute evolution. Evolution will affect larger portions of a population. That's what we want to focus on as evolution. And we want to see lasting changes lasting changes that produce more unique traits, traits that we probably wouldn't necessarily see between one generation to the next. They are the accumulation of many heritable traits. All right, so I just wanted to be careful about that.
This episode of State.
All right, so here's some related concepts to evolution. Coevolution is something we'll talk more about. In fact, you really only need to write down the first sentence here, joint evolution of two or more species, because we will come back and discuss some of the specific relationships that exist in nature that kind of spurn this coevolution. So coevolution is just joint evolution of two or more species. And we've got some interesting examples there as well. Okay. Cool. All right. One more video.
By now, you've surely picked up on the idea that the study of natural history is basically the study of how the world has changed. The study of how we all got here. And of course, the world continues to change all the time. I mean, literally. I'm not just talking about how life adapts or how the climate is changing. I mean, the planet itself as an object is in a constant state of flux because the ground beneath your feet is always moving. So the place where you are right now was not always there. For example, if you're watching me in California right now, then about 500 million years ago, the place that you currently think of as here was actually on the equator. And it was underwater. Likewise, if you're in the UK, then your here was almost at the South Pole, and your next door neighbor was Africa. We know all of this thanks to paleogeography, the study of how the physical face of Earth has transformed over time. And it's this movement of the continents that has driven many of the major revolutions in life's history, its advances and its setbacks, its explosions of life and its extinction events. So if natural history is the study of how we got here, then you also have to understand how here got here. If you want to know how your here got to be where it is, you first have to know where land comes from and how it behaves. And for a long time, we just didn't know. The idea that the continents could actually move was first proposed in 1912 by German scientist Alfred Wegener. He spent years traveling the world collecting geological and fossil evidence to argue that all of the continents were once connected. Wegener was the first to propose the idea of Continental Drift, and the notion was so outlandish at the time that it basically cost him his career. And part of why no one accepted Wegener's theory was that no one in his day had ever seen the bottom of the ocean. Until the mid-20th century, most scientists assumed that the seafloor was basically featureless, like a giant wading pool. But in the 1950s and 60s, pioneering researchers like Marie Tharp and Bruce Heezen began studying the bottom of the ocean. And they found that there was an enormous mountain range running right through the middle of the Atlantic. Eventually, more of these undersea mountain ranges, called mid-ocean ridges, were found in all the world's oceans, and they were all volcanically active. These ridges, it turned out, are where the seafloor is made. Geologists realized that these mid-ocean volcanoes are actively creating the rocky material of the seafloor and spreading it outward in a process called seafloor spreading. But what the sea gives, it also takes away. Far from these ridges, at the edges of continents, researchers also found huge trenches. And here, the dense rocks of the seafloor dive below the lighter rocks of the continents in a process called subduction. So the denser, heavier crust that makes up the seafloor, known as the oceanic crust, moves under the lighter land masses known as the continental crust, just a few centimeters at a time. And as oceanic crust sinks back to Earth's interior, it begins to melt and mix with the mantle. So seafloor spreading and subduction are the two key mechanisms behind plate tectonics, the theory of how giant chunks of Earth's crust, called plates, move around the surface. And together they explain what Alfred Wegener never could. They show us how the continents of our planet come together and break apart. But Wegener was right. Throughout the planet's history, land masses have been joining together to form supercontinents, only to break up again millions of years later. Supercontinents begin to separate when the mantle that's turning around beneath them starts to change, like in direction or temperature or intensity. These kinds of changes, we think, can cause plates that were once pushed together to gradually spread apart. When plates separate, they first create a rift valley, like the Great Rift Valley in Africa. Then as they keep spreading apart, they can form narrow seas, which is how the Red Sea came to be. Eventually, these gaps can open up to become a whole new ocean. That's actually how the Atlantic Ocean formed. This whole process of continents coming together and splitting apart is known as the supercontinent cycle, and it is what has changed the face of the Earth over the eons. Now, you've probably heard of Pangaea, the landmass that contained almost all of Earth's dry land about 250 million years ago. For a long time, experts thought that was the world's first supercontinent. But today we know it wasn't the first, and it won't be the last. One of the earliest supercontinents that scientists have evidence for is called Kenorland. It existed 2.7 to 2.5 billion years ago, toward the very end of the Archean eon. It was made of continental crust that would eventually become parts of North America and Africa. And even though we call it a supercontinent, Kenorland actually wasn't much bigger than Australia is today. Life on Earth at the time of Kenorland was probably mostly single-celled, like the photosynthetic cyanobacteria that were starting to add oxygen to the atmosphere. Then, 700 million years after Kenorland spread apart, another supercontinent began to form, called Nuna, or Columbia, depending on who you ask. The northern reaches of Nuna included land that would eventually become North America and Antarctica. And in the south were the cores of South America and Africa. Nuna existed from 1.8 to 1.4 billion years ago, in the middle of the Proterozoic eon. And it's here where we find fossils of the earliest plant-like organisms, red algae, which lived in a shallow sea in what's now India. Nuna broke apart, but the fragments came back together about 100 million years later. This new continent, called Rodinia, was the first supercontinent that geologists had found existed before Pangaea. And geologists were able to reconstruct Rodinia after they noticed that Labrador on Canada's east coast fit quite nicely into the west coast of South America. Rodinia broke apart about 900 million years ago, and shortly after that, the world was plunged into another long ice age. About 650 million years ago, the next supercontinent, called Pannotia, came together. And here the first animals were found in the fossil record, living in coastal waters from the poles to the equator. First came the mysterious Ediacaran fauna, and then the animals that mark the Cambrian explosion. Animals probably didn't live on Pannotia though, because no fossils have been found in terrestrial rocks from that time. But the land wasn't lifeless, it had likely been colonized by pioneering bacteria, algae, and then the fungi. After Panotia broke up about 550 million years ago, the continents began looking more like the world we recognize. 470 million years ago, in the Ordovician period, the first plants began to live on land. The earliest plant fossils have been found in South America, which was part of the continent of Gondwana. Then, 420 million years ago, in the late Silurian, the first millipedes crawled through the undergrowth on a separate continent just north of Gondwana. And it's known by the catchy portmanteau Euramerica, because it contained parts of both North America and Europe. Euramerica is also where the earliest fossils of insects have been found, about 20 million years later. Then, toward the end of the Devonian period, 365 million years ago, the first amphibians left the swamps to explore the ancient forests that would later form the coal deposits of Europe and North America. And then the earliest amniotes, vertebrates that lay shelled eggs on land, showed up 310 million years ago, right before the formation of the most famous and most recent supercontinent, Pangaea. Pangaea began to form 300 million years ago at the end of the Carboniferous, when North America and Eurasia, together known as Laurasia, collided with Gondwana. And because there were no oceans in their way, animals were free to roam all over Pangaea, which is why similar species are found in areas all over the world in this time. But life there wasn't exactly a picnic. Because Pangaea was so incredibly huge, moisture from the oceans couldn't reach the interior, which made most inland regions pretty much uninhabitable. And of course, making things even less picnicky were two really terrible mass extinctions. First was the Permian Triastic extinction 252 million years ago, aka the Great Dying. It was probably caused by a series of massive volcanic eruptions from fissures in Pangaea in what's now Siberia. These eruptions likely set coal fields on fire and dumped massive amounts of CO2 into the atmosphere and oceans. The super hot air and super acidic rain and seas that followed killed almost everything. 50 million years later, the Triassic came to a close with another mass extinction, wiping out a huge number of crocodile and mammal relatives. This too seems to have been caused by volcanic activity, only this time as North America started to break away from the rest of Pangaea. But among the survivors were the dinosaurs, and as Pangaea broke apart, dinosaurs on different continents became isolated and developed into vastly different forms. The semi-aquatic Spinosaurids, for example, lived on the remnants of Gondwana. Meanwhile, horned dinosaurs like Triceratops mostly lived in North America. Then, after the KPG extinction wiped out the non-avian dinosaurs 66 million years ago, it was mammals' turn. Living on isolated continents like the dinosaurs once did, they too diversified into lots of different and weird forms. Finally, those isolated continents came into contact again in the last 5 to 10 million years, allowing animals to cross new land bridges into new environments, ones we can recognize pretty well today. But of course, things keep moving today just like they always have, at a rate of about 2.5 centimeters a year, in fact. And scientists can predict how the world might look in, say, 50 million years based on how fast the continents are moving and in which direction. So what will future Earth look like? Well, North and South America are both moving west as the Atlantic Ocean continues to grow. Africa is moving north and will collide with Europe, probably forming a huge mountainous plateau, kind of like the Himalayas, where the Mediterranean Sea is now. Australia is also moving north and will eventually smash into the Indonesian archipelago. But beyond the next 50 million years or so, the future becomes harder for us to see. One theory, called Pangaea Ultima, proposes that a subduction zone will form off the east coast of the Americas, closing off the Atlantic and forming another supercontinent like Pangaea in about 250 million years. Another theory, called Amasia, supposes that the Atlantic will keep getting bigger, and that North America will join Europe and Asia at the North Pole. And a third theory, called Novopangaea, envisions a future Earth that's similar to Amasia, but with the Pacific Ocean closed off, as Australia and Antarctica move into the former ocean basin. By then, of course, a quarter billion years from now, our descendants and the other descendants of the modern world will have evolved and diversified to occupy a planet that looks totally different. But they'll be along for the same ride that we're on today, as forces deep within the Earth cause our idea of here to slowly drift, just as it has for billions of years. You and I have been through a lot together today, so I appreciate you sticking around for this whole saga of the supercontinents. As always, I want to
All right, a couple more related terms. So as it relates to Pangaea, which he obviously was just talking about, geographic isolation is a primary cause of speciation. That's a lot of Asian. But geographic isolation occurs when two different populations of the same species end up being separated by some geographic barrier. It could be a mountain range, it could be a valley. We see the Grand Canyon was a source of geographic isolation for certain species. It could be an ocean or a river. Whatever it might be, the two populations become separated, and as a result, they do not have the ability to mate anymore. So over time, based on their new environments where they might be subject to different predators, They might have to find new prey. They might have different food sources in general. They might be exposed to different climates. Over time, this leads to them developing different traits, and so they might eventually become two species. And so this is the process of speciation. This is how new species form. There are a lot of different barriers to the creation of new species. There are both pre-zygotic barriers and post-zygotic barriers. We won't define those in this class, but basically what it means is some barriers exist to prevent different species from having or from mating. Some barriers exist that prevent them from, they don't prevent them from mating. They can mate successfully, but maybe their children don't survive. The offspring don't survive. Or maybe the offspring themselves cannot have children. Okay, so that's the difference between pre-zygotic and post-zygotic. We see different species of birds, for example, that have basically everything in common, but they just mate at different times of the year. So they just never are actually creating offspring. So they're considered two different species because of that. That would be a pre-zygotic barrier. Back. Okay. All right, so now we'll talk about 4 sources of evidence for evolution. The first is fossil evidence. So in the supercontinents that he was just mentioning in that video, we can find evidence that life has existed in different places at different times. So for example, we find alligators in what is today Argentina and Uruguay that we also find in South America. We find plant species in what is today Australia that we can also find in India and in what is today Ethiopia, and even in what is today Bolivia. Same plant species. But of course, there's no way that the wind could have blown those seeds over oceans. So it's actually evidence of the fact that there was at one point a supercontinent that allowed them to be spread across the land. So the fossil evidence is a big source, and it's probably the first source of evidence of evolution that there was. We didn't have, obviously, the ability to analyze DNA when Darwin was doing his work in the 1800s. But we did have the, go ahead. Mm-hmm.
This is one of the this thing for Tectonics that there's fossils and rock evidence of places that shouldn't be connected.
Yeah, they actually mentioned that exact thing in the video. Where did you learn that?
I learned it from the 8th grade.
Okay, good memory. Yeah, fossil evidence, super important. So here's some cool fossils that are pretty historically important. The first fossil on the left is one of the earliest land-dwelling photosynthetic plants. So for a long time, the atmosphere on Earth was not conducive to life. There was no oxygen, or there was very little oxygen in the atmosphere, and it was primarily carbon dioxide. Because of this lack of oxygen, we don't see life on land that needed oxygen for respiration. But we do see eventually, and this is one example, organisms that were able to do photosynthesis and that were doing anaerobic respiration. So they were creating energy without the use of oxygen, but through photosynthesis, they were producing oxygen. So over the course of about the first 1,000,000 years of Earth's existence, we didn't see any life on land. And then as these plants started to find their way onto land, they filled the atmosphere with oxygen, and this made land much more conducive to life. We also see there a fossil of a fish with fins joined to the body by a single bone. So this is a precursor to tetrapods. Of course, our limbs are joined to our midsection by singular joints. And so this allows us a much larger range of motion. And this fish is probably a common ancestor of most tetrapods, like you and I. And then we also see the Lotali footprints on the right, which are about 3.7 million years old. And they were left by some animal that was able to walk upright, some vertebrate that was able to walk upright, and could be considered an ancient ancestor of humans. So interesting stuff. All right, so of course, he kind of mentioned this in the video talking about subduction, but when we're looking at rock layers, which ones will be the oldest? Where do we find the oldest rock layers? The ones that are furthest down. So in this case, that would be number 4, and then the youngest rock layers are at the top. They're most near the surface. So, okay, that was pretty simple. The second evidence of evolution is comparative anatomy. This is when we actually see similarities between the organs and skeletal structures of organisms, even across various phyla and classes. Convergent evolution is when different species begin to look more and more alike. So, for example, there have been several different species of crustaceans that have developed qualities of a crab. Yeah, that's the classic example. In fact, there's even a name for it. I can't remember exactly what it is, but comparative anatomy is when, of course, there's some type of common ancestor, and even as evolution takes place, we still see different species maintaining some of the anatomical structures of that common ancestor. So this is kind of divergent. They're moving apart from one another, but maintaining similarities. Whereas convergent is they were already pretty different, but they're becoming more alike. So we see Tiktaalik in this image. I think I showed you Tiktaalik in an image on Tuesday. Tiktaalik lived around 375 million years ago and represents a transition between fish and early tetrapods. So because of this, we've been able to study the fossils that have been found of Tiktaalik, and we see some similar anatomy, including wrist joints that, of course, mammals have that many other organisms in different phyla do not have, rib bones, lungs, an ear region. So, of course, Tiktaalik didn't have ears that could process audible stimuli, but does have an ear region, as well as a mobile neck. So if you imagine a fish does not have the ability to move its head without moving its entire body. But of course, that is an important feature for mammals. We have a neck that allows us to move our heads without needing to move the entire body. Yes. All right, so quick scientific name review. This very first name right here, Tiktaalik, which of the 8 levels of scientific classification is that? That's the genus, good, thank you. And then Rosi, what would that be? That's the species, thank you, Sully, yeah. So we always include the genus and the species in a scientific name, good. Another very important type of comparative anatomy is called homologous structures. So this image really depicts homologous structures quite well. We've got 4 different types of mammals here: a human being, a dog, a dolphin, and a fruit bat. and we're looking at the bones of their limbs. We can see that humans have a bone in our upper arm that's called the humerus. We see that same long bone appearing proximate to the trunk of these other animals. Of course, it looks different in dolphins and fruit bats, but it's that very first bone that is connected to the trunk of the body. We also see two bones that appear more distal, meaning further away from the trunk of the body, that connect that long bone to the hand, essentially. In humans, we call the bone that's on the side of your thumb the radius, and we call the bone that's on the side of your pinky finger the ulna. In humans, we have bones that make up our wrist, and we call those bones the carpals. But we also see those same bones that allow mobility in dogs and dolphins and in fruit bats. The metacarpals in humans constitute the bones of the hand. So when you've heard someone say, I broke my hand, you're kind of like, wait, what? What did you break in there? Well, there are 5 bones in there that constitute really the palm of your hand, and they're called the metacarpals. And then lastly, the phalanges would be the fingers. But we still see those phalanges apparent in dolphins, fruit bats, and dogs. So it's pretty cool. All of these are mammals, of course, and this hints, again, this is suggestive of a common ancestor. We maintain these structures. Yes, they have grown to be different to allow for, different types of survival, but they still exist. Okay. Is anybody still writing? No. This is a cool one. I think it's cool. Vestigial structures are anatomic features that used to have a function, but that no longer do have a function. So they've lost all function in the living species. So for example, and we're going to watch a video of this, we've got muscles behind our ears that, if you actually move your eyebrows up and down, you can feel your ears moving. Or sometimes if something scares you, you might feel your ear move a little bit. Those muscles, of course, are there because our ancestors probably needed to have the ability to move their ears. If some of you have pets at home, you've probably seen your pet's ears perk up when they get excited or when they hear something off in the distance. You might see them move towards the source of a sound. So those same muscles that are doing that for your pet, we also have them in our heads. They're just not as developed, and our ears obviously aren't as big. Wisdom teeth are also a vestigial structure. The appendix, if you've ever heard of someone having an appendectomy, meaning that they had their appendix removed, probably because it was infected or it burst, they can go on about their life after having it removed as if nothing happened, because the appendix does not provide an immediate use to us. It doesn't have any real function in our digestive system. And it's there because it is a vestigial structure. So you might ask, why hasn't evolution gotten rid of these structures if they don't do anything for us? That's a good question. I think the consensus thought is that they are not so costly that they provide maybe a detriment to our survival. But maybe if given a few more hundreds of thousands of years, we might see these structures no longer apparent in humans. That would be kind of frightening.
What they were trying to say is like they're going to lose how small and useless it is.
Yeah, maybe. Then again, natural selection is not acting as much on human populations as it might in other organisms, right? Because we are not subject to our environments as much. We have temperature controlled rooms. We can go to the grocery store. Right, exactly. The third type, and probably the most important type of evidence for evolution is comparative biochemistry. And this is literally analyzing DNA or proteins to observe similarities between two species. So we can literally count how many nucleotides do two species have in common, or how many amino acids do two species have in common. And the more common, or the more that they have in common, obviously, the more similar they are. Mm-hmm. All right. And then the fourth and final evidence of evolution that we'll discuss is comparative embryology. And this is basically exactly what it sounds like, comparing embryos. And the embryos, of course, are the earliest stages of development in utero. So does anybody think they know what any of these species are, just looking at this early embryo stage, early embryonic stage? That's a safe bet. Which one, though? Okay, what makes you think that? Okay. So Lance is saying that he thinks this one? He thinks that this embryo right here is a human being. Okay, and he's citing its size as well as the specific size of this cranial structure here. Anybody else think they know what any of these embryos are? Okay. There is a species of bird, yes. Okay, so you think this is the bird? Let's see. Does this make it more obvious? These are embryos in middle stages of their embryonic development. So now we can see that hind limbs are developing and forelimbs are developing in some of them. Okay. So now Lance is saying he thinks this green one is some species of bird. What about these two on the far left? Okay. I'm highly impressed. Okay. So this is a fish on the far left. There's a salamander indeed. I'm impressed by that. The green one is a tortoise. The yellow one is a chicken. That's the bird. And then Lance, you were right about the human too. So 2 out of 5 is not bad. All right, but the point is, when you look at them in their earliest stages of development, it's almost impossible to see any real differences, at least with your naked eye. Of course, if we were to do genetic testing or even just chemical testing, we would be able to see some differences. But yeah, even human beings. What Lance is saying for those at home, even human beings, he's surprised, well, not surprised, but would you say you think it's, he said he thinks it's cool that there are gill slits, even in human beings. Of course, we do not live underwater, but we do develop in utero in what is called amniotic fluid. So before the development of the umbilical cord, which delivers oxygen from our mothers to us, we need some way of getting oxygen, and we need gills to do that. So it's yeah. Yeah. So babies do often have pretty bad cases of hiccups in the moments immediate after birth, but they also cry because it's the first time that they're breathing. Prior to that, like I said, you get your oxygen solely from your mother's umbilical cord. And if some babies do actually have issues where they get fluid in their lungs because they try to take their first breath too early. And of course, there is no air inside of the amniotic sac. So it's interesting stuff. Yeah. Not a comfortable feeling. Okay, one more review question. What happens in the early stages of embryonic development to create different types of cells and different tissues and different organs? What's the name of that process that creates different cell types? Any thoughts? Any guesses? What'd you say? Give you a hint. It creates special types of cells. When you see the answer, you're going to be like, okay, that was a pretty good hint. Thank you, Abigail. This process is called cell specialization. Yeah, so this is related to gene regulation. We talked about this in Unit 5, I want to say, maybe even Unit 4. But certain genes are activated, other genes are deactivated. All of our cells have the entire copy of DNA, but they have different genes activated, and this gives them different specials or specializations. One more video, and then we'll be done.
Your body is a temple, but it's also a museum of natural history. Look closely and you'll see your body is a temple, but it's also a museum of natural history. Look closely and you'll see parts that aren't there because you need them, but because your animal ancestors did. No longer serving their previous function, but not costly enough to have disappeared. These remnants of our deep history only make sense within the framework of evolution by natural selection. With your arm on a flat surface, push your thumb against your pinky and tip your hand slightly up. If you see a raised band in the middle of your wrist, you've got a vestigial muscle in your forearm. That tendon you see connects to the palmaris longus, a muscle that around 10 to 15% of people are missing on one or both of their arms. It doesn't make them any weaker though. There's no difference in grip strength. In fact, it's one of the first tendons that surgeons will take out so that they can use it in reconstructive and cosmetic surgeries. You can find the palmaris longus across mammal species, but it's most developed among those that use their forelimbs to move around. In primates, that means the muscle is longer in lemurs and monkeys and shorter in chimps, gorillas, and other apes that don't do a lot of scrambling through trees. It's not the only leftover muscle that we've got. Look at the 3 that are attached to our outer ear. We can't get much movement out of these muscles, especially compared to some of our mammal relatives who use them to locate the sources of sounds. Presumably, this would have been quite helpful for early nocturnal mammals. In humans, you can still detect the remnants of this adaptation with electrodes. In one study, researchers recorded a spike of activity in the ear muscle cells in response to a sudden sound. Not enough to move the ear, but detectable. And you can probably guess the location of the sound based on these results. It came from a speaker placed to the left of the study subjects. So this is their left ear subconsciously trying and failing to pivot toward the sound. You can see another futile effort by our vestigial body parts when you get goosebumps. When we're cold, tiny muscles attached to our body hairs contract, pulling the hair upright, which causes the surrounding skin to form a bump. For our furry mammal relatives, the raised hair increases the amount of space for insulation, helping them stay warm. Birds can do this too. You've probably seen a puffy pigeon on a cold day. Adrenaline is one of the hormones involved in the body's response to cold temperatures, and it's also part of the fight or flight response. So it helps some animals appear larger when they're threatened. And it may be why surprising and emotional turns in music can give some people goosebumps. And then there's our tail. At the end of our spine are a set of fused vertebrae. Some people have 3, some people have 5. We call it the tailbone. It now serves as an anchor for some pelvic muscles, but it's also what's left of our ancestors' tails. Every one of us actually had a tail at one point. When the basic body plan is being laid out at around 4 weeks of gestation, human embryos closely resemble embryos of other vertebrates, and that includes a tail with 10 to 12 developing vertebrae. In many other animals, it continues to develop into a proper tail, but in humans and other apes, the cells in the tail are programmed to die a few weeks after they appear. Very rarely, though, a mutation allows the ancestral blueprint to prevail, and a human baby will be born with a true vestigial tail. The most adorable vestigial behavior is the palmar grasp reflex, where infants up until they're about 6 months old have this incredible grasp on whatever you put in their hand. There's a similar reflex for their feet. I wanted to show you this great piece of footage from the 1930s where they demonstrated this behavior. These babies are only 1 month old, and you can see that their inner monkey can support their entire weight.
All right, so we've got 5 minutes left. There is an exit ticket. Now, I've noticed, in fact, let me just go ahead and pull it up. And those. All right. So if you look here, we're only completing about 70%, or at least in Unit 7, we only completed about 70% of the warm-ups and exit tickets. In Unit 6, it was 78%. In Unit 5, it was 83%. So we're trending downward, folks. The warm-ups and exit tickets, as I will remind you, are often taken directly from past exams. So those questions are excellent practice for the EOC as well as for each of our unit exams. In fact, I think they're good study tools as well. And I've mentioned that a lot in the past. I just wanted to remind you, we should still be doing these warm-ups and exit tickets. Use the time that you're given so it doesn't feel like you know, night before the exam, you need to go in and do them all. Okay. So 4 minutes, that should be enough time to do the exit ticket. It's only 5 questions. All right, folks, one more day tomorrow. I will see you then. I hope you get some good rest tonight. Looks like we're all in need of it. Good rest and fresh air, that'll do the trick. Sunlight. See ya. I know. I wish it were.